a session on systems biology, I guess it sort of causes me to rethink about the project and ask if we're doing systems biology, and I guess the answer is yes to a certain extent. When I think about systems biology, I think about sort of the classical issue of trying to understand how, for example, metabolic pathways work within a single cell. So where the scale of complexity is restricted in a sort of geographical sense, it's microns, although complex in a biological sense. In our case, our system is the natural environment as well as agricultural systems. And so it has a different kind of scale. We think about function over hundreds of thousands of square kilometers. And our system is complex in the sense that we're looking not just at a particular plant species that I'll introduce in just a minute, but co-occurring species and abiotic factors like soil and climate, and trying to understand how these influence the function of the system. So basically our goal, or one of the objectives, is to understand gene function, which genes contribute to the function of organisms, in natural systems where coevolution and evolution has been going on for millennia, and in agricultural systems where, in fact, probably links to those natural evolutionary processes are broken and new ones have been established through breeding. Okay, so we have, our project has two main focuses. One is to, and our species of interest is chickpea, by the way, that I'll introduce in just a minute, a legume. One focus is to understand the impact of domestication, which in chickpea is about a 10,000-year event, on processes related to nitrogen and nitrogen fixation, the comparative advantage of legumes, nitrogen fixation. And the other one is to understand to what extent there are alleles left in the natural system that might be useful for addressing some current agricultural challenges in chickpea. And in our case, we're interested in tolerance to climatic extremes, heat, cold, and drought amongst them, but also nitrogen fixation, disease resistance, soil type adaptation, seed nutrient density, primarily those areas. So a broad set of objectives. We have, to go with those broad set of objectives, we have a large team and in fact an international consortium. The main part of the project is funded by the U.S. Agency for International Development. This is involved in looking for useful alleles in wild systems and moving them into cultivated. I guess in a sense it's an attempt to re-domesticate the species. We have funding from the National Science Foundation, which does upstream research to understand the dynamics of the natural systems, which isn't of interest to an applied agency like USAID, but I would assert, and I'll hopefully show you some data, that the more you understand about the biology of the natural system, the better you'll be able to make decisions about how to use that information in cultivated systems. We also have co-funding from the Grains Research and Development Corporation that funds a large program, multiple laboratories in Australia, as well as from the government and a growers organization in Canada, and funding from the Global Crop Diversity Trust, who has a particular interest in using wild genetic diversity for crop improvement. So it fits nicely with our goal. Our focus is chickpea. In a broad sense, it's the second most important pulse legume. Depending on where you are in the world, if you're in India, it's the most important pulse legume. It is relatively closely related to a species named Metacago truncatula that my group has worked on now for about 25 years. And so for us, this gives, in a phylogenetic sense, a species that has close proximity with a lot of functional data and very good genomics. And so we leverage between Metacago and chickpea. Chickpea is also, you can look at chickpea's importance in a different way, and this just shows the top 10 chickpea producing countries in the world, and it makes a couple of important points. First of all, far and away the largest producer, consumer, and it turns out importer of chickpea is India. Another important point here is that the yields of chickpea on a per hectare basis vary substantially around the world, and this is suggestive that there are opportunities for improvement, potentially through genetic gain, but also through agronomic practices. And I would add that agronomic practices could include the use of improved inocula for nitrogen fixation, for example, and it's something I'll talk about a little bit later. Now, our target country, we begin working at chickpea center of origin, which, by the way, is in southeastern Turkey. But our target country, at least for the purposes of USAID, is Ethiopia, for a number of reasons. But in sub-Saharan Africa, chickpea, the largest amount of chickpea by any single country is produced in Ethiopia. It's responsible for about 40% of all of Africa's chickpea production. It's interesting from an evolutionary perspective because it's one of the historical focuses of domestication. The two places outside of the Middle East, outside of Turkey, where chickpea was early on domesticated or used as a crop were India and Ethiopia. So there's a potential for long-standing evolution through breeding as well as potentially coevolution with rhizobial symbionts in these secondary centers of diversity. Ethiopia is at risk from climatic extremes, which fits nicely with the focus of the USAID project on climate resilience. And there's a unique dependence of the human population in Ethiopia on chickpea, in particular because for religious reasons they fast for a significant period of time during the year where they don't eat meat. And during that period of time, which is about 2 months, they have a high dependency on protein nutrition from plants, and chickpea in particular. So a range of reasons why Ethiopia is important. So I'm going to spend a little bit of time talking about sort of a base resource that's important to both the NSF project, which says how do natural systems function, and the USAID project, which says what attributes of natural systems might be useful if we could get them into cultivated systems. The base resource is to make a collection of material from these natural environments. So one question that's important is how do you sample accessions, new genotypes, with adaptations to environmental extremes, one of our focuses. And our answer to that question is we want to survey across the full geographic range of the species, and we want to survey diverse climatic and soil conditions. In our case, we have a pretty narrow geographical region, and so altitude ends up being a driver of climatic differences. We sample about a threefold difference in altitude, from slightly more than 500 meters in elevation to slightly more than 2,000 meters in elevation in southeastern Turkey. And along with those climatic differences become differences in rainfall gradients, the seasonality of rainfall, temperature gradients, humidity, and so on. And we know from work that we've done characterizing these in situ environments, there are big differences in soil types. And we believe those soil type differences are probably driving plant local adaptation, but also influencing the structure and function of microbial communities, as well as co-occurring plant species. So the second question, aside from how do you find environmental differences, is how do you sample across big geographical regions? So the region is about 100,000 square kilometers, and this encompasses the full known range of the wild progenitor of chickpea, which is Cicer reticulatum, and a sister species, Cicer canispermum. Cicer eritinum is cultivated chickpea. So the species, the geographical range is slightly east of the Euphrates. It begins eastward of the Euphrates River up to the border with Iraq and Iran, which is apparently a physical barrier to the species. There's no herbarium records of Cicer, of either of these wild species eastward of these mountains. The northward physical barrier, a few hundred kilometers north of the Syrian border, that also defines the northern border of the species. And southward into Syria you have deserts, and so the species doesn't exist. Another way of putting that is the geographical range is quite narrow and well delimited, and that's where we have to search. So how do you find the species in those areas? We began by going into herbarium records as well as into the international gene banks and the multilateral systems, taking metadata about the species, coming up with GPS coordinates, going into the global climate data sets, and pulling out environmental data variables, and modeling the likelihood that a given area within southeastern Turkey would be a good candidate for finding new populations. And with this information in mind, this search image, we got in several cars, and we spent several person years, but in fact about 8 months, about 6 months in total, driving around southeastern Turkey. And this is headed out on a dirt road, and further in the distance, that water that you see is the Tigris River. And so we drove for about 2 months in 2013 and another about 2 months in 2014. We're getting ready to head back into the field for about 2 months this year. We found 50 sites, over 2,000 plants, most all collected as DNA, some collected as seed. Once that material is in hand, now we can ask, what did we find when we collected that material? So when we would go into a site, sampling strategy is important. We want to understand the variation within sites as well as among sites. So when we go into a site, we'd mark every plant with a red flag, and you can see them way back in the distance. So we'd collect up to 100 individual accessions per site. We'd GPS tag them, we'd collect DNA. We'd come back later in the season when the plants are maturing and collect seed. And while we were there, we'd collect soil from multiple points at each site so we could do chemical analysis. And we would conduct environmental sampling. So we're collecting as much data about these natural environments as we can. From the DNA that we collected, all GPS tagged, we can sequence it and get an understanding of the spatial scale of the distribution of genetic and genomic variation. And a first sort of analysis of that is simply to look at population structure, in this case based on allele frequency data using a program called structure. And what it tells us is that amongst the 1,069 accessions that we collected in 2013, of course we can discern the two species, Cicer reticulatum and Cicer acanispermum, and we can discern a number of distinct populations. Most populations correspond to single sites. Some populations are distributed across multiple sites. And with this now we can look at genetic differentiation, FST, and other kinds of parameters that allow us to ask how much diversity is there within a site and how much do different populations differ from one another. I'm not going to present that analysis, but you can see that it's the beginning of trying to describe diversity in a systematic way. Another important question is, okay, we went out and we spent a lot of time driving around. We collected new material, but is that new material different than the material that's currently in the gene banks? It's certainly quantitatively different. It's a couple of order of magnitudes more than what was currently available. So if you take the GPS coordinates for the historical collections that are in the gene banks, the majority of that material, but not all of it, but the vast majority derives from a single region just around a town, near a town called Mardin in southeastern Turkey. And it comes within about a 4-kilometer radius. In other words, the gene banks are predominated by a single population of wild chickpea. And they have not sampled, they do not contain representatives of a large number of additional populations, new genetic variation, and new phenotypes that we can see when we grow them in the greenhouse are missing from the international collections. And so we've expanded both genetically and phenotypically the variation of these of the international collections. The material is presently the gene bank in Izmir, Turkey at AARI, and it's available with the use of an MTA. At each site, in addition to collecting genomic DNA and sequencing those genomes, we have collected environmental data. We can go into the global climate data sets and collect information about the sites generally, but we wanted to understand the microheterogeneity of climatic variation within a site. And so we planted these small sensors. They're about the size of a nickel. They go into the soil. We plant them at the depth that a seed might be. And every 4 hours they collect information about moisture and temperature, and they do that over a 2-year period. We go back, pull these sensors out of the soil, stick them into our tablets, download the data, put them back in the field, and then come back later to collect them. And an example of the kind of data that can come out of this, in this case you can take the data about temperature and you can look for temperature triggers that should affect development, germination and flowering, for example, so we can express the information as cumulative degree days. This kind of information tells us that the sites differ substantially in the kinds of environments that should be driving local adaptation, which is an important issue for us. So as an example of how we're using climate data. At each site we also collected soil. We took soil cores. You can see them in the bottom right-hand side of the screen here in these plastic tubes. Each of those cores don't just represent soil, they also represent root rhizospheres of chickpea or soils that don't contain chickpea. So we can use these soil cores both for chemical and biological analysis. And so the chemical analysis that we've done in this case in Diyarbakir is to look at a range of soil parameters. And we know that the soils differ substantially, and those differences correlate with the genetic structure of the populations. I'm not saying that soil type differences are causal to population genetic differences, but there are correlations. And I won't go into this, but we have a large amount of information about soil types. And we also take that soil and we have fractionated the soil into a range of different fractions to begin sequencing the metagenomes, the total microbial communities that are associated with the plants, to ask questions like what is the scale of, what's the geographical scale of taxonomic variation of microorganisms that are associated with chickpea? That's one question, biogeography. Another question is chickpea structuring the microbiome, and does it do it in reproducible ways? So a range of questions that we're going to be asking in these natural systems to look at microbial communities. And this project is, part of the project is just underway. We're in the process of sequencing about 800 metagenomes collected from those natural environments. In parallel, we've gone into the nodules where nitrogen fixation occurs, and we've isolated rhizobia. We developed living collections and we've sequenced the nodules directly from which we can assemble genomes. And I'm going to tell you a little bit about that. Okay, so that's the base resource. The project I'll tell you about now is what we've done in the NSF project, where we have the beginnings of some data about the biological systems. Remember, the NSF project asks, what are the dynamics of nitrogen and nitrogen fixation in the natural systems, and how have they shifted upon domestication? Now, the motivation for this project actually comes from observations made in soybean and in pea. And in both soybean and pea, there's evidence that domestication, human selection, have impacted nitrogen fixation, and that they've potentially made nitrogen fixation less efficient. Another type of data that suggests that looking at nitrogen and nitrogen fixation could be of interest is that crop legumes differ in the amount of residual nitrogen that they leave year to year. And even within a single species, if you look at a range of different cultivated instances, you'll see very big differences in the extent to which biological nitrogen fixation is satisfying the nitrogen demand of the plant. In other words, there's lots of variation, some agronomic, some biological, and trying to get a handle on that could be of interest. And if domestication has impacted this in negative ways, there's an opportunity potentially to go back and try to fix it. So these are all hypotheses, of course. We have some data to support them. The project's just about a year old, so we're making progress. Here are a few observations that we've made. And in each case, we want to try to understand the genetics that underlie these. We know that wild and domesticated species differ in the way that they respond to nitrogen in the soil. Think about it as fertilizer. Wild species do not use fertilizer nitrogen at levels that are normally found in a fertilized field. They're insensitive. They use symbiotic nitrogen very well. Cultivated species respond very well to nitrogen in the soil. And you can see this both in terms of plant growth as well as in the total N balance in the plant. We also know that there are big differences in the partitioning of root and shoot biomass and the way that's done in wild systems compared to natural systems. Wild systems have, in a nutshell, larger root systems relative to total biomass than cultivated systems do. And interestingly, in Arabidopsis, where this has been well studied, the partitioning of biomass between roots and shoots is a nitrogen-driven process. So it's of particular interest to us. We also know there are differences in the number of nodules formed on the root systems between wild and cultivated plants, looking at multiple different accessions. And we know this is controlled by a single major locus that differs between cultivated and wild. So in each of these cases, we'll spend the next few years trying to understand these properties better and identifying the genes. The place we've made more progress in the first year of the project actually is looking at the structure of the microbial genomes, the symbionts. And I want to tell you about that. So we started a few years ago, well, we just a few weeks ago, we finished sequencing of about 100 genomes, microbial genomes, symbionts, collected from Turkey. And we collected from the same sites that I described earlier, where Cicer reticulatum, the immediate progenitor of cultivated chickpea, or Cicer echinaspermum, the sister species, occur. And we observed that there are two different species of Mesorhizobia that are associating with these two different legume species. Cicer reticulatum associates primarily with an organism known as Mesorhizobium mediterraneum and a closely related species known as Mesorhizobium temperatum, everything in blue here. These are the organisms that dominate in the nodules of Cicer reticulatum in the wild. Conversely, organisms that dominate in the nodules of Cicer echinaspermum, a closely related species in the wild, are a different species of Mesorhizobium, an organism called Mesorhizobium ciceri. And in greenhouse experiments, we can show that, in fact, these two species, it's not simply a consequence of the reticulatum sites having the temperatum organism in the soil and the echinaspermum sites having the ciceri organism in the soil. They actually distinguish differently between the organisms. There are some genetic differences between these two species. So we did an experiment a couple of years ago with Rajiv Varshney's group here at ICRISAT. In this case, we asked whether cultivated Cicer eritinum and the two wild species that I just introduced you to, reticulatum and echinaspermum, if you put them into agricultural soils where there should be a complexity of rhizobia, do they select different organisms from the soil? And so we planted the plants at the end of the season. We pulled up plants. We isolated several thousand nodules. From those nodules, isolated DNA, and then we resequenced directly from about 400 nodules. And we could ask whether the plants are sampling differently. So I made two observations initially. First, in the fields at ICRISAT, just behind you, there are two species of Mesorhizobia that associate with chickpea. The minor species looks like one of the two species that we found at the center of origin in Turkey, Mesorhizobium mediterraneum. So one of the natural symbionts is in the field behind us. But the dominant symbiont is a new organism that hadn't been observed to nodulate chickpea before. It's an organism called Mesorhizobium pluriferium. It's been observed in acacia and a few other legumes, but never on chickpea. So what is it doing on chickpea? I'll answer that question in a second. We also observed that the cultivated species and the wild species planted into the soils that contain these two organisms are selecting differently from those pools of organisms. The cultivated species is selecting more frequently for Mesorhizobium pluriferium, and the wild species are selecting, at least reticulatum is selecting more frequently for the Mesorhizobium mediterranean group. In other words, there are differences in host specificity as expressed by the host for the organisms in these agricultural fields. Okay, so those are two things. Two species of symbionts, and the hosts are selecting differently. Now within Mesorhizobium pluriferium, because we had a whole genome sequence data, we could actually look at phylogenetic structure, and we observed that there were 8 different subgroups or phylogenetic clades within Mesorhizobium pluriferium. It's not a homogeneous entity. But more importantly, if you take any one of those subgroups and look at gene content and ask how homogeneous is a subgroup within pluriferium, you find something that, at least initially, was rather surprising, and that is that the symbionts are massively diverse. So you can do what are called genome accumulation plots. In this case, it's one of the pluriferium subgroups. We had 30 sequenced genomes. Take any 2 of those genomes and annotate them per genes and ask how many of the genes in genome 1 are also in genome 2. You come up with a core genome that defines those 2. And then the genomes the genes that are unique get added together, and they assign what's called the accessory genome. And you can do that with 2, 3, 4, all the way out to all 30 genomes. And when you do this, you find that although any given genome has close to 7,500 genes across the set within this one clade of pluriferium, only about 2/3 of those genes are the core genome, and the accessory genome, which is the fraction of the genome that's not common to the set, is about 3 times the size of the core genome. In other words, the organisms are extremely diverse, and from the standpoint of nodulation and inocula, we think about these things as single, relatively coherent species. The fact is they're nothing of the sort. They're massively variable. This is not unique to chickpea symbionts. It's been observed in E. coli now almost 2 decades ago, and that is that bacteria have well-conserved core genomes and highly variable accessory genomes. An open question is whether the differences in the accessory genomes are impacting the efficiency of nitrogen fixation. Because if they are, then we need to be concerned about which particular strains are nodulating our hosts. Okay, so that was an example of what's happening in Patancheru in the field right behind us. But there's a bigger question, and that's if you look at chickpea globally, how diverse are its symbionts? And so we started a project to collect from 3 sites of historical cultivation outside of the center of origin. So chickpea, again, is native to Mesopotamia. About 6,000 years ago moved to India, about 3,500 years ago to Ethiopia, a few thousand years ago to North Africa. Our study site is Morocco, and quite a bit more recently to Australia and North America, which are recent sites of chickpea cultivation. So what do the Mesorhizobium populations look like there? And I don't have the full answer yet, but I have enough of an answer to tell you there's something interesting going on. If you ask that question for the strains that are collected and then sequenced from those different regions of the world, what you find is there are a range of different Mesorhizobium species nodulating chickpea, not just Mediterranean and Semi-Arid that we found the center of origin, and not just pluriferium that we found in the field behind us. And those Mesorhizobium species are always more closely related to Mesorhizobia that nodulate other hosts and not chickpea. So the group of symbionts that nodulate chickpea is polyphyletic. You can see that in the core genome. Now, interestingly, if you take a look at the genes that are necessary for symbiosis, the so-called nod genes, or the genes that are involved in nitrogen fixation, you find those genes are monophyletic. There are 2 different histories in the genomes of chickpea symbionts. There's a monophyletic history that tracks from the Middle East for a portion of the genome. There's a polyphyletic history that probably corresponds to the genomes of organisms that are in the places where chickpea has come to be cultivated. And I'll describe that more in just a second. In fact, it turns out that the monophyletic fraction of the genome is a large collinear region that represents about 10% of the genome, and it is a horizontally transferred section of the genome flanked by transposable element-like sequences. So if you're a chickpea symbiont, you have a polyphyletic core genome. You have a monophyletic 10% of the genome that drives symbiosis. So another way of looking at the domestication history of nitrogen fixation is from the bacterium's perspective. And the domestication history is about the horizontal gene transfer of 10% of the genome consistently into new microbial genomes that are pre-existing and characteristic of the new agricultural situations. So there's a piece of the genome that's hopping around as chickpea has been domesticated in the bacterial genome. So how could that help us? Well, now you can think about the possibility of strain development for inoculum. We imagine that the core genomes that are variable are important to soil type adaptation, while the horizontally transferred portion of the genome is essential to be a chickpea symbiont. So what is that horizontally transferred region of the genome doing? Well, it's actually well known. The horizontally transferred region of the genome encodes genes that are involved in host range. And we observe that chickpea selects differently from these different organisms. In the bacterium, that core region of the genome, among other things, codes for a group of genes called the nodregulon, that's responsible for the biosynthesis of a signal molecule called nod factor, an N-acetylglucosamine oligomer that has various modifications on it. And if you want to be a symbiont of chickpea, you have to make nod factor with a very particular structure. How do you do that? You acquire the mobile fraction of the genome that specifies that particular molecule. Okay? And so it's been moving from genome to genome, giving new organisms, new species, the capacity to nodulate chickpea. Another fraction of the genome that's contained on the transferred region in some microbial symbionts and in these particular chickpea symbionts is another group of genes that actually regulate host specificity. And these are the so-called HRP genes and effector genes, and they're best characterized by people who study disease resistance. And they encode a molecular machine that's responsible for delivering bacterial proteins from the cytoplasm of bacterial cells through a tube into the target cell. Their purpose is to modulate signaling in the cell. In pathogens, they try to stop signaling for defense. In symbionts, they modulate host response so symbiosis can occur. At least this is the emerging paradigm. And we find that both of these genes that can specify host range are present on this transmissible region of the genome. The important thing about this is now we have a lever to begin to work in the bacterium. But because of the long history of research on disease resistance and nitrogen fixation in the host genome, in chickpea and other legumes, we also know the host genes that are the counterparts to these bacterial products. The counterpart to the nod factor biosynthesis regulon are a series of genes that are receptors for nod factor. They bind that oligomer and they specify nodule development. And both in Medicago, our model system, and in chickpea, there are gene families. The transcription of these receptors is induced by the ligand itself. It's relatively well characterized. I don't need to go into the detail, but we have good candidates in the host that are the counterparts of that signaling molecule that's moving horizontally. And similarly, there are good counterparts in the host that can recognize the function of the effector proteins. And these are things involved in disease resistance. So we have candidate genes that we know are moving in the bacterium, and we have a bunch of candidate genes in the host that are potentially specifying host range differences that we observed in the field. And although this sounds like a lot of basic science, if I still distill it to a practical application, you can think now about going into the chickpea genome and to the genomes of its microbial symbionts and seeing if you can come up with better combinations of organisms that are soil type adapted, but also allow for efficient symbiosis and modifying both sides of the equation. Now, I want to close by sort of stepping back one point and saying that the long-term goal of the project, of NSF, it's basic biology, but of USAID, it's trying to use these populations, these plant populations that we've collected for crop improvement. And to do that, we have to build populations. So we're building nested association mapping panels where the target are 5,000 NAM rills that are normalized for phenology, and a corresponding set of advanced back-cross integration lines, both of which can facilitate gene discovery and marker assignment to traits, as well as developing material that will initially be pre-breeding and then eventually lead into breeding activities. We have a range of different partners. I'll just mention the main institutions. UC Davis is the lead. We have partners at Florida International University in the States and at the University of Southern California. We're working with Vincent Videz here at ICRISAT, and historically we've worked with Rajiv Varshney. In fact, a lot of the symbiosis work that I talked about derived from a collaboration with Rajiv. We have collaborators in Turkey at Harran University and Dicle University, as well as at national programs in Turkey. We collaborate with a range of groups in Western Australia and in Canada at the University of Saskatchewan, and we have two groups of partners in the national programs and at Addis Ababa University in Ethiopia. And I'll stop there.
Thanks, Doug. I think we have time for one quick question. Yeah.
Thank you very much for your high quality research work. Again, proving the quality of UC Davis. Yeah, and my question is, in the process of domestication, you told this nitrogen fixation has come down. So, along with nitrogen fixation, what genes might have been lost in the process of domestication, particularly adaptability genes?
What genes might have been lost during domestication?
Adaptability genes, means stress resistance.
I'm sorry, I heard the first part of the question, but not the last part. But, so your question is what was lost during domestication, is that correct?
Yeah.
Well, I mean, it's a good question, and we want to know the answer to. So right now we have phenotypes, and the question is what genes underlie those phenotypes. We know that host specificity has changed. We think there's good reason to believe that it involves these candidate genes that are the receptors for the bacterial signals, but that has to be proven. In the case of nitrogen responsiveness, differences in nodule number, and so on, we'll spend the next 4 years trying to find the genes. So I don't know the genes. We know there are phenotypes that differ and that those phenotypes are interesting. Yeah.
Okay, many thanks, Doug. So